Honorable laureates, distinguished colleagues, ladies and gentlemen, welcome to the Nobel Lectures in Chemistry 2022. This year's Nobel Prize in Chemistry represents a most beautiful synergy between tremendously strong and eye-opening concepts and very important discoveries. Through the concepts of click chemistry and bioorthogonal chemistry, amalgamated and boosted with the establishment of enormously useful reactions, our three laureates have given us means and tools to see beyond the intricate complexity of chemistry and address important challenges while being able to connect complex molecules. Our three laureates, Carolyn Bertozzi, Morten Meldal, and Sharpless, found truly efficient solutions to what is often very, very complicated. Bioorthogonal chemistry, giving us high selectivity and reactivity that work in the most complex environments. And click chemistry, giving us highly efficient and robust reactions that work almost everywhere all the time. With this chemistry, many important challenges in chemistry and related fields could finally be met. And, in a sense, the laureates opened up a floodgate, the floodgates of a Hoover Dam filled with unmet needs, which very quickly resulted in a wealth of new discoveries across many different areas. These discoveries have undoubtedly made the strongest impact on science and our society. For example, leading to the deciphering of complex biological systems and disease processes, as well as the development of functional structures and tailor-made materials for advanced use in various applications. So, let's start with our first laureate. Carolyn Bertozzi was born 1966 in Boston, Massachusetts, USA. She was awarded a PhD from the University of California at Berkeley in 1993, and is now active as the Ann T. and Robert M. Bass Professor at Stanford University, where she is also an investigator of the Howard Hughes Medical Institute. So by that, please join me in welcoming Dr. Bertozzi up on stage.
Thank you. This is the honor of a lifetime, to say the least. And it's my great pleasure to tell you about the work that we did to develop bioorthogonal chemistry and to apply this new chemistry to imaging of cell surface glycans and many other applications related to that. Let me start by just introducing one of the basic tenets of the field of chemical biology, which is my field. And one of our tenets is that there are many unmet needs in the biological sciences and in medicine that drive the development of chemical technologies. And bioorthogonal chemistry is very much one of those technologies that was inspired by an unmet need in biology. And at a very high level, bioorthogonal chemistry was developed by my coworkers and myself when I started my career at UC Berkeley. And the fundamental goal was to invent a new type of chemistry that could be performed in what I think of as the most complex reaction vessel you could think of, which is the living organism. And this flies in the face of the traditional ways in which we think about chemical reactions. When we do reactions in our chemistry laboratories, we're typically using this kind of a reaction vessel. It's a round-bottom flask where the chemist can control every single parameter of that reaction. By contrast, if your goal is to do chemistry in a living system, as was our goal, You're no longer in the reaction flask, and you no longer have control over all of the parameters. Instead, you might be doing your chemistry in a live cell, in a cell culture dish, for example. And there are a lot of interesting problems you could solve by doing chemistry in the live cell. For example, to attach imaging probes to biological molecules that you want to study in their native habitats. And that's a very complex environment in which to think about chemistry. But even more complicated is the chemistry you might want to do in living organisms, like a laboratory mouse, or, as I'll show later in this talk, the zebrafish, which is a model for development of vertebrates. And probably the highest level of complexity where one might want to do a chemical reaction is the human. And in fact, as you'll see, bioorthogonal chemistry is actually now being tested in human cancer patients for the purposes of drug delivery. So for all of these kinds of reaction vessels, we knew that we needed a different brand of chemistry. And so we coined this term, bioorthogonal, to describe the qualities that you would need to do chemistry in these living systems. So our definition is that bioorthogonal reactions are reactions among components that neither interact with nor interfere with a biological system. And one way to think about that is that there is an enormous reactivity space that's described by biology. I like to think of biological reactivity space as a galaxy of chemicals within a much larger universe of chemicals. And it's a large galaxy, but there's a boundary around it. And somewhere outside of that biological reactivity space is the bioorthogonal reactivity space, a different galaxy with different chemicals, functional groups that you would not find in a biological system, but that will react with each other regardless of being surrounded by all of those biological functionalities. So that's the type of chemistry we'll be talking about today. Now, as I mentioned, chemical biologists often develop new technologies in response to an unmet need in biology. And in my lab, the unmet need that we became interested in was to study changes in cell surface glycosylation. That's another word for the complex carbohydrates that decorate the surface of all of our cells. And even back in the 1990s, there were decades of literature showing that when cells undergo a transformation of their state, there are changes in the patterns of cell surface glycans. And sometimes those changes in cell surface glycosylation are associated with disease. For example, it was already known back then that when healthy cells transform into a cancerous state in the context of a tumor, there are cell surface glycosylation changes that often include an overproduction of a particular sugar that's known as sialic acid. And it's one of the building blocks in the complex carbohydrates on the surface of your cells. And we had a fairly straightforward objective, which was to develop a technology that would allow us to profile the abundance of sialic acids on healthy cells compared to cancer cells, and to develop a strategy for molecular imaging of changes in sialic acid in order to study cancer. But at that time, there was no technology available to image cell surface sugars, including cell surface sialic acids. So that was the unmet need that we were focused on solving. How do you image a sugar? Well, you have to do it in a different way than you would image other biomolecules, like proteins or nucleic acids. For those other molecules, there were some good technologies available at the time, but for sugars, there was not. And during my postdoctoral years, so this takes me now back to around 1991 or 1992, I was thinking about this problem. the need to image the cell surface sugars. And I happened to attend a conference in Southampton, England. And this was a serendipitous moment for me because I heard a lecture by the late Professor Werner Reuter, a very well-known German biochemist at Free University Berlin. And his lab had been studying the biosynthetic pathway that produces sialic acids and delivers them to the cell surface. And what he had observed, and he presented this at this conference, is that one could feed cells a chemically altered version of a metabolic precursor of sialic acid that's called N-acetylmanosamine. And normally, N-acetylmanosamine has an acetyl substituent here at this pink position, where this R group would be a methyl group. And what he found is that one could make conservative changes to this R group, just extend the side chain out by a few carbon atoms. And those subtly altered N-acetylmanosamine derivatives would be subject to enzymatic metabolism inside the cell, which led to the production of an altered sialic acid residue, which was then incorporated into the complex carbohydrates on the surface of those cells. So you feed the cell a chemically altered sugar, and they produce a chemically altered sialic acid. And when I saw that talk, a thought came to mind, which was that maybe we could put a chemical group that's reactive at that position. And once that chemically reactive group appears on the surface of the cell, we could chemically attach an imaging probe. So that was one of the research proposals that I crafted and submitted when I applied for a faculty position at UC Berkeley. And here's a cartoon that's almost literally right from my job application from 1994, where I proposed a two-step process for imaging cell surface glycans. The first step would be to feed the cell or the organism, or maybe the human patient someday, that chemically altered simple precursor sugar. And that sugar would be metabolized by the cell, converted to the cell surface product, where this chemical group X, and I was agnostic about X at that time because I wasn't quite sure what X would be, but some chemical group X would come along for the ride. Now it's presented on the cell surface, and we would do a chemical reaction with an imaging probe that had a complementary functional group Y. And the hope was that X and Y would react with each other, form a bond, and that would attach the probe to the sugar, and we would now be able to image the sugar. But there was one problem with this cartoon, which is that we would need a chemical reaction between two functional groups that could proceed in a living system, in cells, in animals, the natural environment of these cell surface sugars, which means that the chemical reaction would have to be bioorthogonal. And this was the problem that was the genesis of bioorthogonal chemistry. So the reaction that we started with, and which ultimately was the first bioorthogonal chemical reaction, was a derivative of a very well-known chemistry from the German literature of the previous century. This is the classic reaction of triphenylphosphine with what turned out to be a very important functional group. You'll hear a lot more about this in the other talks this morning, which is the azide. And phosphines and azides will react with each other to form an adduct called an azaylide. This was a chemistry first reported by Hermann Staudinger over 100 years ago, 1919. And in fact, Professor Staudinger won the Nobel Prize in Chemistry about 40 years after this initial report. Now, phosphines and azides do not exist in biological systems, and they react with each other very selectively without any interference by the biological system. So on its face, this reaction, as I've shown it, looks like it could be a bioorthogonal chemistry. But there was one liability with this classic Staudinger reaction, which is that the azaylid product is not stable in water. And if you're doing reactions in living systems, water is your solvent. There's no choice. So we had to solve this problem of the hydrolytic sensitivity of the azaylid. And one of my graduate students, Eliana Saxon, who joined my lab around 1998, came up with the solution to the problem. She made a simple modification of triphenylphosphine, introducing this methyl ester. So now, when the phosphine reacts with the azide to form this azaylid, the nitrogen atom, which is nucleophilic, can attack the ester in an intramolecular reaction, very rapidly forming this cyclic product. So now, when water ultimately hydrolyzes this phosphorus-nitrogen bond, the amide bond that had formed in that intramolecular reaction keeps this product together as a ligation product. So this variation on the classic Staudinger reaction, we called the Staudinger ligation. And this was the first example of a bioorthogonal reaction. And here is Eliana. I found this old photograph. The day that she got her first big result, where she showed the Staudinger ligation could be performed on cultured cells in a dish with no harm to those cells. And she came into my office and she showed me the data, and I remember thinking, I think this is a really big result. But this was very early in the days of my young lab. So once we had this chemistry worked out, we then demonstrated that we could introduce the azide into a variety of different cell surface sugars, all of which are interesting in their own ways. So for example, I mentioned that imaging sialic acid was one of our initial goals. And so we showed that we could introduce the azide on the side chain of N-acetyl-minosamine. And when fed to cells and metabolized inside the cell, the corresponding azidosialic acid would appear on the cell surface, primed for reaction by the Staudinger ligation. And likewise, we figured out how to put an azide into a sugar called fucose, which is also an important sugar for various biological systems. And in addition, we showed that we could put azides into N-acetyl-galactosamine, or GalNAc, using this azidoacetyl variant of the parent sugar. And each of these sugars we chose because there were some interesting biological stories behind them. Another big moment in the lab came when two of my former graduate students, Jen Prescher and Danielle Duby, demonstrated that the Staudinger ligation could be used to attach probes to sugars in living mice. This was another big moment in the lab because it was the first demonstration that a bioorthogonal reaction could be so well tolerated by a living system that you could do the chemistry in the mouse with no toxic side effects. And in fact, Jen and Danielle, I'm so privileged to say, are with us in the audience today. And here is an image from this paper they published showing that they could inject into mice azidoacetyl-minosamine. It would get metabolized by the cells in the body of the animal, and the azidosialic acid would appear on the surface of those cells. Then, in a second step, they would introduce a phosphine probe set up for the Staudinger ligation. And in the body of the animal, the phosphine would find the azide, undergo the reaction, and form this product, which we could then detect in a variety of ways, including with fluorescent-tagged monoclonal antibodies. Another major achievement by these fearless students who were willing to turn the mouse into a reaction flask. But we knew that there was more work we had to do in reaction development. And the reason was that the Staudinger ligation, for all of its amazing qualities, was a rather slow reaction at biological temperatures. It's a bimolecular reaction, which means that it follows bimolecular reaction kinetics. And so hopefully the students in the audience will recognize that for such a reaction, the rate is equal to an intrinsic rate constant times the concentration of the two reactive partners. And in a biological system, like a living animal, we learned some things that are important. For example, we learned that the concentrations of these reactants were probably going to be very low. The animal is metabolizing these reagents as soon as we put them in the body. This is not a reaction flask in your fume hood. And there's only so much of a molecule you can introduce into an animal because of issues like solubility in aqueous systems or non-specific toxicities for molecules that really don't belong in the animal's body. So given the limits on the concentrations of these two parameters, the intrinsic rate constant of the reaction is very important if you want the reaction to proceed at a reasonable rate. And for the Staudinger ligation, which had a second-order rate constant around 10 to the minus 3 per molar per second, we discovered that this was a little too slow. This reaction took hours to proceed, sometimes a day. And there's a lot of biology that takes place on a much shorter time scale than a whole day. So we were interested in alternative bioorthogonal reactions that would leverage the azide as a reactive partner. Now it turns out that azides can also undergo what we call 1,3 dipolar cycloadditions with alkynes. This is another chemistry that was well established in the previous century. And Professor Rolf Huisken from University of Munich was one of the major practitioners of this chemistry, who did a lot of mechanistic work to describe how this reaction proceeds. And like the classic Staudinger reaction, this chemistry seemed to us as being almost bioorthogonal, because like the azide, alkynes are also quite rare in biological systems. We don't have them in our bodies, as far as we know. And yet these two will react with each other. Now the reason I say this is almost, but not quite, bioorthogonal is because this reaction is very slow at room temperature, much slower even than the Staudinger chemistry. Which is why when people do these reactions in the fume hood, in the chemistry lab, they generally have to reflux the reaction at high temperatures, like 100 degrees C. So at biological temperatures, we thought of this Huisken chemistry as just being too slow for our needs. Now in 2002, the world was changed. And that's because my co-laureates, Professor Barry Sharpless and Professor Morten Meldal, discovered a mechanism for accelerating the rate of the azide-alkyne cycloaddition. And this is what is now almost synonymous with the term click chemistry. And as you will hear from their glorious lectures after mine, the addition of a copper catalyst changes everything. And suddenly, this reaction is incredibly fast. It's over in minutes. And it is now by far one of the most important reactions in all of chemical biology, and very much worth recognition by a Nobel Prize. Could this solve our problem of kinetics? And the answer was not quite. The problem was the copper catalyst was just too toxic for cells and live animals. But it was kind of on the right path, and it got us thinking about other mechanisms by which one might accelerate the rate of the cycloaddition, but without the need for a toxic metal catalyst. Now, like many of us, I teach sophomore organic chemistry, introductory organic chemistry. And when we teach this class, we always have a lecture on the concept of ring strain. And here are the two textbooks that I used to use to teach this class when I was at Berkeley, both written by Berkeley faculty, Peter Volhart, Clayton Hethcock, and Andy Streitweiser. And in these books, you will see a description of what happens when you make ring systems that have uncomfortable bond angles. These strained ring systems can cause functional groups to have very different reactivities. So for example, when you put a double bond into a three-membered ring in this form or in this form, a lot of strain energy gets pent up in that ring. And if you can react those pi systems and release the strain, that can accelerate the rate of a reaction dramatically. So I talked with my students about whether there was precedent for an azide alkyne cycloaddition with a strained ring system. And Nick Agard, who is sitting here in the audience today, went to the library, which used to exist, and he found this amazing precedent. Back in 1961, Georg Wittig, another Nobel laureate in chemistry, reported on the reaction of cyclooctane, the smallest of the stable cycloalkynes, which has 19 kilocalories per mole of strain energy. And when Wittig and Krebs, his coworker, combined cyclooctane with phenylazide, you can see from this statement in German, the reaction proceeded like an explosion to form the corresponding triazole. So here was an azide alkyne cycloaddition with no catalyst that basically proceeds at room temperature without the need to heat it up. So based on that precedent, Nick and then Jeremy Baskin, who's now a professor at Cornell, and he's sitting here in the audience today as well as several other coworkers, went to task to try to develop cyclooctane-based probes for what we called a copper-free click chemistry that we could use in living systems. And to make a long story short, they spent several years building cyclooctanes with different kinds of modifications. And what we learned is that the cycloaddition of these cyclooctanes with azides can be tuned in terms of reaction kinetics over several orders of magnitude. So we had a lot of control levers on the rates of these cycloadditions. Just to give a few examples, in the upper left is a very simple cyclooctane, which would react with a model azide with a second order rate constant that was kind of on par with the Staudinger ligation. So that was our starting point, but we knew that we needed to enhance the rate by a few orders of magnitude. And so Jeremy Baskin had a big breakthrough in this project when he showed that addition of 2 fluorine atoms next to the triple bond would increase the reactivity almost 100-fold. And this reagent, which we gave the acronym DIFO for a difluorocyclooctane, became one of the first important reagents for imaging sugars in live systems, as I'll show you next. But just to complete the slide a little bit, we could enhance the rate yet another order of magnitude by fusing benzene rings on either side of the cyclooctane, which increases the ring strain even a little bit more. And then the ring could be shrunk down to a 7-membered ring as long as one of the atoms in the ring was a larger atom like sulfur. And one could get yet another boost in the reactivity, where now we have a reagent that's 4,000 times more reactive than our parent compound. So this was a really fun period of time in the lab because we felt like we were returning to our roots as physical organic chemists, doing the kind of structure reactivity correlation studies that were popular 100 years earlier, but that had fallen out of fashion in the new millennium. And yet here we are invoking some of these classic concepts to tune a reaction for biological systems. With these new reagents in hand, Jeremy Baskin and another coworker, Scott Laughlin, took on the challenge of imaging sugars in live animals. And the animal that they focused on was the zebrafish, which is a very popular model organism to study vertebrate biology, with the benefit that zebrafish are translucent. So they're a wonderful organism for fluorescence imaging studies. You can see all of their organs under a microscope as they are alive. And so Jeremy and Scott did experiments where they would take early embryos of zebrafish developing in a dish. They would feed these embryos azido sugars, which would get metabolized. And once those azido sugars appeared on the surfaces of the cells of the animal, they would do the copper-free click chemistry reaction in the live animal with a dye-faux probe, introduce the fluorescent dye, and now we could image the sugars for the first time in a living animal. It was a very dramatic moment in the lab. And they did some experiments where they would pulse these embryos at different time points with different azido sugars and map out sugars appearing in different cell types. So for example, in one experiment, they would do one round of labeling, and then any unreacted azides still present in the fish, they would quench with a reagent that is a phosphine reagent in a classic Staudinger reduction. Then they could pulse the embryo with more azido sugars that would appear at different time points and label them with different colored dyes. And we could create fish that basically had all the colors of the rainbow, where the different colors represented different types of cell surface sugars that appeared during development at different time points. Just to give you a sense of what these images looked like, up top is the head of a roughly 5-day-old zebrafish, which has had different sugars labeled at different time points with different colors. And you can see how they're distributed. And below that is basically a round of cell division from an earlier embryo. So here's the intact early embryo. Each arrow is pointing towards cells that are undergoing mitosis. And if you expand the image and take a close-up look at cell division, what you can see is that a single parent cell, I'll wait for it to loop, duplicates its chromosomes, segregates them as the cells divide, and you can see the cell dividing through the portal of the DNA, which there are good tools for imaging, and we used one of those tools here. You could see the cell division through the portal of the proteins, which were also easy to image back then, and the proteins that we're imaging here are located on the membrane of the cell, and now it's dividing into two daughter cells. But for the first time, we could also see cell division through the portal of the cell surface glycans. And that was another first-in-class achievement by these incredibly fearless group of students. And in this image, we were quite struck to see how the glycans build up and concentrate right at the junction as the cell is completing a round of mitosis and contributing to the segregation of these daughter cells in a way that was previously unknown. So after this point of time, the field really took off because it turns out that once you show that you can use chemistry to image sugars in a biological setting where the animals are alive, lots of people came out of the woodwork and asked us if they could use our reagents to image all kinds of other kinds of molecules that were also difficult to study. And so here's a pie chart on the cell showing roughly the breakdown of all the different types of biological molecules. And over the course of 4 or 5 years, and this brings us up to about 2010, there were groups that showed that they could use the azide as what we call now a chemical reporter for virtually every type of molecule on the pie chart. They could put azides into amino acid side chains and label proteins and use bioorthogonal chemistry to image those proteins in living systems. They could put azides into nucleosides and label the oligonucleotides and use bioorthogonal chemistry to image those molecules. They could put azides into lipids and track those lipids around in living systems using bioorthogonal chemistry. So the applications of this chemistry in the research setting expanded quite rapidly into lots of different areas of biology. Another big development around that time was expansion of the toolkit of reactions that are available for bioorthogonal chemistry. And so we started our work with the azide, and the cyclooctane partner became a really good combination for chemistry in living systems. But later, other groups discovered that tetrazenes can be bioorthogonal reagents reacting with strained alkenes like this cyclooctene or this cyclopropene. And now that toolkit continues to grow, and I would put the challenge out to students in the audience here. If you come up with a new idea for a new bioorthogonal chemistry, this could have tremendous impact. Because while we now have a handful at our disposal, there's always a need for more options in this space. It's a great challenge for someone who's starting out their career in organic chemistry. Also, the applications expanded. So 20 years ago, we developed these chemistries as tools for basic research in the laboratory setting. But over the last 10 years or so, there's been considerable activity in the translation of this chemistry for new medicines and new diagnostic strategies for human disease. And so I thought I would mention that there are now a number of biotechnology companies that use bioorthogonal chemistry in a variety of settings. A very popular application is for making antibody drug conjugates. And these are important medicines for the treatment of cancer. As well, there are companies that are using bioorthogonal chemistry to make new vaccine conjugates. And some of these are in, right now, a phase 2 clinical trial. And then I have a company, as a disclosure, which uses bioorthogonal chemistry to make targeted degraders of extracellular molecules. And I'm very excited to be an advisor of a company called Shasky, which is performing bioorthogonal chemistry in the body of cancer patients as a mechanism to target chemotherapies very selectively to the tumor tissue and to keep those chemotherapies away from healthy tissues. So it's a very exciting time for me to see how the chemistry came from a very modest idea in the research lab aimed at imaging sugar molecules into a very generally applicable tool which people are using to make new types of medicines. Finally, I want to make one point, which I think is important, because despite all of these exciting applications, I still think the most important lesson from the story of bioorthogonal chemistry has to do with the importance of curiosity-driven basic science. And those of us who are scientists are familiar with what people often describe as a tension between basic science and applied science. And funding agencies go through various phases of interest in applied science versus basic science. But bioorthogonal chemistry tells the story of how important curiosity-driven basic science can be in order to realize these applications. Because I'm pretty certain that the chemists on whose shoulders we built bioorthogonal chemistry, and by that I mean Hermann Staudinger, Rolf Huisken, Georg Wittig, when these chemists reported on the curious reactivity of azides, phosphines, and alkynes, they would never have foreseen that someday I would stand here and show you fluorescent zebrafish. They couldn't have known that. They made observations about exotic molecules and they published them purely based on curiosity. And here we are 100 years later making use of these chemistries in ways that we hope will benefit human health. So in order to get to fluorescent zebrafish and imaging of sugars, to imaging of tumors in human cancer patients, and to making new medicines, you need to have that foundation established by the basic science. So with that, let me thank the bodies of people that basically made this moment possible. First and foremost, the Royal Swedish Academy of Sciences and the Chemistry Nobel Committee. Thank you for changing my life. The Nobel Foundation for the amazing festivities of this week, which are just ramping up. My institution of origin, UC Berkeley, which we often call Cal. This is where all the work was done that I presented today. So I owe a great debt of gratitude to the students and to the faculty at Cal that gave me my big break and that did all this beautiful work. And my current institution, Stanford, is very supportive and is helping me try to translate some of these reactions into human medicines. And then finally, the people who financially supported this work from the very beginning, which is the National Institutes of Health and the Howard Hughes Medical Institute. The most important slide of all is this one, Because none of this happens without the intrepid students and postdocs that occupy the academic laboratory. And this is my current group at Stanford. This is how we had group meetings during COVID. We had to rent a drone to get this picture. And after the Nobel Prize announcement, my students organized a Zoom party. And these are all the faces that we could get on Zoom on short notice from all over the world, and some of whom I'm presenting this work to today. Thank you very much for the work that you did leading to this moment, sitting there in the second row. My family is here. Thank you very much for all the support you have given me since my moment of birth. And thank you very much to this audience for coming today. Thank you.
Thank you so much for that fantastic presentation. It's time for our next laureate, Morten Meldal. He is a native of Denmark and was born in Copenhagen in 1954. In 1986, he was awarded a PhD from DTU, the Technical University of Denmark, located in Lyngby, just a bit north of Copenhagen. And he's now active as professor at University of Copenhagen. So by that, please join me in welcoming Dr. Meldal up on stage.
Dear colleagues, friends, Dear colleagues, friends, ladies and gentlemen, I write A Leap from Shoulders of Giants, and I would like to start in reverse order of the talk we just heard by thanking all the giants that are behind the work that I'm going to talk about today. So my heroes, some of them, Klaus Bock here in the audience, together with Wüttrich on NMR, structural studies on carbohydrates and peptides, Hüsken and Woodward and other people that are doing cycloaddition reactions, Prigozhin, Bach and Gray on quantum chemistry, Paulsen, Lemieux, and I think that we should also mention Shepard and Merrifield on the peptides and carbohydrates, and then Furka and Lam and many more on combinatorial chemistry. These are really the people that we built on in our research. So before I dive into the lecture, I would like to thank also the Nobel Foundation and the Nobel Prize Committee of Chemistry, as well as the Royal Swedish Academy of Sciences for this absolutely unique event. And I'm very appreciative and very thankful for being able to stand here today and very proud to share this moment with Carolyn Bertozzi and Barry Sharpless. I also thank all the colleagues at Carlsberg Laboratory and University of Copenhagen for all the things that they have done during the years in development, discovery, development, and application of this click chemistry. And finally, I want to express my thanks and love for my family who have supported me over the many years of science, and in particular my wife, Phaedra Saint-Hilaire, my son, Ajani, who is here today and is celebrating his birthday, my daughter, Anna, and Anna's mother, which is not here today, Sandra Davolio. Finally, I would like to express the thanks to my late parents, who would be so proud to be here. And my perception of nature, and in particular the underlying chemistry, is a testimony to me of the absolute beauty and the complexity of the nature and our very existence. So nature has been with me since early childhood, and I'm very grateful to my parents for dragging me through forests and making collections of rocks, butterflies, insects, and so on, so that they actually spurred my interest for science. And later in life, I chose science as a subject for my education. And at a certain point, I had to choose between chemistry, physics, and computational sciences. And I chose, fortunately, chemistry, because today, to me, chemistry is everything. There's nothing around us that is not chemistry. You are chemistry, I'm chemistry. All our surroundings are chemistry. And in fact, we will need chemistry very much if we have to solve all the challenges that are ahead of us. The global challenges will need chemistry solutions, and we need our young people to learn chemistry from the very point. So from very young, we need to go through this process and learn about chemistry. So after completing my study, I went into carbohydrates and peptides, and I spent some time at the university, and then I ended up at Carlsberg Laboratory. And at Carlsberg Laboratory, I combined glycopeptides, or peptides and glycans into glycopeptides in order to start study glycoprotein functions. And during this process, I also established the Spock Center funded by the Danish National Research Foundation. And the reason was that there was a great need for quantitative chemical reactions at that point. So we had a great focus on QCT reactions, as we called them at the time. And this was within carbohydrates, peptides, and proteins using combinatorial chemistry. And for those who don't know what combinatorial chemistry is, I would like to give a short explanation. So we have some small plastic beads that we use for combinatorial chemistry, and they can contain different starting points. As you can see, they are orange and green and purple. And now we can couple reagents with quantitative chemical reactions to these beads and form one combination. We can then mix these beads up into a complete mixture, and then we can redistribute them into these wells and couple a second reagent. Now, each of these small plastic beads is a container for biological assays, as well as they contain only one chemical compound. So if we have 3 Lego blocks and 3 steps of reaction here, we'll get 27 compounds, or 3 to the third. If we have 5 steps and 5 compounds, we get 3,000 compounds, or 5 to the fifth. If we have 20 compounds and 5 steps, we'll get 3.2 million compounds. So you can see this is an exponentially growing process of generating compounds at very large numbers. And we therefore needed these very perfect reactions in order to do that. So during this time, this is showing how we can do this in practice. The beads are in here. We do the coupling reactions, or mixing them, and we prepare them for another coupling reaction, and we take around this cycle many times. At the end, we can take these beads into a biological assay with enzymes or binding biomolecules, and then we can identify the active compounds on those beads that are showing a sign of activity. So this is, from the combinatorial chemistry side, very important that we can identify those compounds here, like needles in a haystack, and we are using mass spectrometry to do that work, but this is another story. Because here at Carlsberg Laboratory, we did the discovery and the multifunctional molecules by click that I will describe. Click reactions, what is it actually? The click reaction, and then I have some applications I would like to present to you. During this very active period from 97, a lot of people were working in the lab on establishing N-aluminum reactions, amination reactions, and also we tried to make keto-alkynes for branching peptides and making disorder reactions on peptides. And this here is a method for peptide synthesis that we developed where we can synthesize extremely difficult peptides that cannot be synthesized in any other way. It's based on the A site as a protecting group for amines and the acid chlorides for coupling. Very high activity of these compounds in the coupling reactions. So one of our very skilled PhD student, Christian Tornø, who is here in the audience, had a project in which he had to take alkynes, make the copper acetylenes, and then react those with the acid chloride so we get these acyl alkynes. That did not happen in his reactions. He fought a brave battle in order to make this reaction work over several months. Nothing would work, and the reason was that this irritating byproduct was formed all the time. It was such a pain, but when we looked in detail into this irritating byproduct, we found that this was actually the triazole, and it was formed quantitatively even in the presence of this very reactive acid chloride. And you can see there's no starting material left, and everything is converted in a short period of time. So Christian investigated the compatibility of this reaction with all the peptide functionality that you have in proteins. And he presented this work at the APS in San Diego here. So thank you very much, Christian, for your work. Carolyn alluded to a universe. I'll just talk about a toolbox that pharmaceutical industry is using with all these molecules that we make in pharmaceutical industry, and the many problems that are associated with solvents, with excess reagents and so on in this toolbox to form the well-known drugs that we are taking today. Also in material sciences, you have these kind, for example in polymer synthesis, you have these kind of tools that are not very clean and not very green chemistries. And then you have a very small toolbox today, as introduced by Carolyn already, which contain the QAC reaction and Carolyn's two reactions that you see here. This little toolbox contains, as you also saw from Carolyn's slides, only 3 reactions. And it's 20 years ago we started this research, which means that it's extremely difficult to find reactions like this. So we have this little toolbox that's completely orthogonal to the chemistry going on out here. And that means that we can take things from this big toolbox and combine it like molecular Lego. So I would like to show you what we can do with click chemistry in a putative example where we have formed this guy here. He is good to kill cells from the inside by apoptosis. Then we have this monoclonal antibody that can locate tumor tissue, and we have a cell-penetrating peptide that can take the other guys with them into the cell if they are connected. So now we take these 3 guys and we put them into a solution of copper-1 ions. And once they're in there, they find each other on the surface of the copper and they couple and make the triazole. So we can do that even in with completely deprotected proteins and in water solutions. This is quite unique. And we get this guy here, and now this guy, he can find a cancer cell like this one, and he sits on the cancer cell for a while, and the cell-penetrating peptide does the work, carry them inside, and now the apoptosis can work, and if we're lucky, the cell is dead. So click is good, and click is green. So click can be carried out in water, and click doesn't lose any of its material. If we are clicking these two guys together, you can see we can find all the atoms in the product. So atom economy is very important here. So this is a kind of reaction that you would really like in order to combine two large parts in a new pharmaceutical. But what is really click? We start out with an alkyne, and this is a big bag of electrons. It's full of electrons in orbitals or balloons or whatever you would call them, here sitting on the surface of the alkyne. That's negative charge. It would love to see a copper 1 atom and bind it here to this orbital. That acidifies the alkyne out here. So the copper atom takes the place of the proton on the end of the alkyne. That's still not reactive enough. So we take another copper atom and take it in. It comes in there, sits on the electrons, and it peels off the electrons from the alkyne. So we get a positively charged alkyne, so to speak, with empty orbitals that are looking for electrons. And where can we get the electrons from? Well, one place we can get them from, which fits almost exactly with the faces of these balloons here, is the A side, and here comes the A side, and it fits exactly to form what we call a metallocycle. The metallocycle that loses first one copper atom to form a triazole and a second copper atom, so that we get the triazole product that now connects this functionality with that functionality. I think this is what we got the Nobel Prize for. So the fact is that we can use this reaction to couple very complex molecules, because this reaction is compatible with all the different stuff you can see here in these two parts, and form a triazole very selectively. And now we have a peptide that has a functionality of some recognition and some cell penetration. But there's one problem, and that's a redox issue. The problem is that the copper 1 actually very easily becomes copper 2. And this is a reaction that we cannot prevent it, but we can sort of help out by reducing back copper 2 to copper 1 here. This is fine if we have unsensitive molecular structures, but as soon as we have highly susceptible functional groups, as in macromolecules, this is really an important issue. You have to get rid of all the reactive oxygen species, as they are called. That's these guys here. They can damage your protein completely. And the way to do that is to avoid oxygen, avoid redox cycles, and use protective ligands for your copper 1 catalyst. Once you do that, you can actually do with 0.01% of copper 1. So it's a real, true catalytic reaction. And I would like to present some of Christian's work where he has seen if we can use this to do inhibitors for enzymes, use this click reaction. So we are having these combinatorial libraries of beads. On these beads we have synthesized by the combinatorial method I showed you a very large number of compounds which have variations in these 5 positions. And what we are looking at here is a sandfly that transmits Leishmania mexicana, something that you die from. So if we can prevent that, that would be a very good thing. And we can now look at the beads. And because we have here a substrate which is fluorescent, when we remove that part over here, we can look at enzymes that cleave this substrate when this is not an inhibitor. But if it is an inhibitor, we cannot cleave this substrate, which is attached to the same reactor, the same small microparticle. And here's one that is an active inhibitor in the presence of all the inactive molecules here. So Christian isolated a number of those and found the best one. And this is the best one, 870 nanomolar inhibitor. We weren't very excited about that because this looks like we have much better inhibition. So when we looked at this, we thought maybe this leucine actually is the one that's binding in the first site just on top of the active cysteine in this protease CP28. So Christian synthesized the same structure as you have up here, but now with the mass spacer that we use for analytical purposes here. And now we get a very decent inhibitor of 76 nanomolar. So we thought it would bind like this with the triazole just on top of the active cysteine, but in reality it bound like this on a much larger surface and interacted very strongly with the entire binding site here. And that's how this inhibitor actually works. That was the first example. The second example I will show you is on the melanocortin receptors. These are very important receptors, and we found out already in the beginning of the click era that we could do macrocyclizations to maintain structure in peptides and proteins. So here we have a cyclization to give us 21-membered ring structure, and this ring structure happened to present these three pharmacophores in just the right way for interaction with the melanocortin receptors. So we have here the three pharmacophores, and they interact with these 5 receptors that are very important receptors. They are involved in obesity, in growth and puberty, in sexual dysfunction, memory, pigmentation, and many more. So here you see one of the receptors in a membrane, in a lipid membrane of a cell. And if we now activate with a ligand here from the outside of the cell, we'll get a response that is transmitted to the nucleus to express proteins. And that is a function of the receptor. So here we have the G-coupled protein sitting on the inside of the membrane, sending the signal to the nucleus. When we come with a ligand up here in the top, these two helices will change conformation and transmit the signal. So we tried to see what is it that happens with this transmission. So we did these 17 compounds with different components here on these positions. And we could do that very readily because we had the click reaction to form the cycle. So now we are binding the ligands up here in the top, and that affects this conformation of these two helices, sending a signal to the nucleus. And then we, instead of the proteins that normally are expressed there, we express the green fluorescent protein, which give us green cells, and we can measure the amount of activation of the receptor by the fluorescence intensity. So that is what you see here with increasing amounts of activation reagents. You can see that the cells become more and more fluorescent. And we get these kind of curves of activity out of it. And the difference between these curves and these two is 300, which means that we were able to get a 300-fold activity on the MC4 receptor compared to the MC3 and MC5 receptor. And this MC4 receptor is the one that is involved in obesity. So we would really like to see if we can use that as a starting point for development of medicinal products towards obesity. In our third example, we are talking about the guy you see here. This is a Japanese horseshoe crab. It's a very special creature. It has been on the Earth for 400 million years, and the reason is the innate immune system that this fellow has. It's a blue blood that encapsulates the bacteria and kills it with these antimicrobial compounds that you see here. This is beta hairpins that are linked together with two disulfide bridges. So what we did was to take this compound and then substitute the two disulfides with triazoles. So we were able to cyclize this in two positions with completely unprotected peptides at the same time, getting these structures very cleanly. And we could then, with the help of Charlotte Gottfredsen, who is here in the audience, analyze that these are actually forming these kind of cannonballs where two of these goes together, hiding away all the lipophilic stuff and being positively charged on the surface. So we have these cannonballs and a membrane that is negatively charged. The positive charge on the cannonball locates them on the surface of the cell, and we get a pore, and through this pore all the contents of the cell will pour out until there's nothing left in the cell and it dies. So that is how this works, and we tried it out on different bacteria to see if our compounds was just as active as the natural one. Here's the result: E. coli, Staphylococcus epidermidis, and Salmonella typhimurium, and Bacillus subtilis. Our compounds was just as active as the natural antibiotic in 3 of the organisms here. In the 4th one, that had obviously developed a kind of resistance to this antibiotic. There was no activity, but our compounds were very active, probably because the Bacillus had developed some sort of disulfide reductase elimination of this antimicrobial peptide. So click is good also for antimicrobial peptides. And in the last example I will show you today, we use this for the clicking of 2 halves of a protease. Proteases are notoriously difficult to express, and they are difficult to express both because they usually can process themselves, so they will kill themselves during the expression process, and also they would kill the host cell because it would start hydrolyzing all the proteins that are inside the cell. So this is a real difficulty, and usually you have to do great protective measures in order to get that expression to go. So the unhappy coli here, we had to do something. So we divided the protease in 2 in a loop, and we have 2 halves of the protease now that are expressed, and we express them with the so-called amber stop codon suppression technology, where we get an alkyne and an acide incorporated in the protein. And now we can just ask the coli, is it happy? It was very happy. So we got large amounts of these 2 building blocks, and we could connect them. Here you have the positions of the alkyne and the acide. We could connect these 2 like that. And now they are just forming the structure in solution without connection. Then we could do the click reaction here by adding a copper-1 ion. It comes in and forms the triazole, and now we have a fully active protease, actually a little more active than the natural protease, as you can see. So this is on hydrolysis of this fluorescence quench substrate. So we are able to use this click chemistry for all sorts of bridging purposes. We can do protein structures, simply replacing disulfide bonds, also in completely unnatural proteins. So there's a lot of opportunity for these click reactions in the future. So on this note, I would like to come with some important last words. I'd like to emphasize the importance of serendipity in research, as you have seen. That was quite important. I think that all great discoveries really have an element of serendipity in them. Because if it was obvious, you would already have done it. And the other thing is that great discoveries are not done behind the office desks. They are done in the laboratory. Having an idea, making very creative experiments, having a keen observational eye on what's actually going on, you will see that there's always something odd happening because nature is very, very complex. Yeah, and I think that could actually be an inspiration to make funding in a different way. Maybe we could do funding so that the researcher was free to decide exactly where to go at any moment. Simply by looking at the performance over the last couple of years and see if this is worth funding into the future. That would be much, much better and much, much cheaper than the way we are doing it today. So the Nobel Prize in Chemistry is important, more important than ever. It inspires, justifies, and provides direction in this challenged world. Chemistry is omnipresent. In fact, it's everything, as I said, it's you and me, and everything else in this world. And chemistry is more than an education, in my opinion. I think we should try to make chemistry a general education for the young people. In particular, I think that we have the possibility now to take our young people and teach them chemistry at a very early point because we have unique abilities to do animation, to show them a world of images of chemistry at a very early point where they can actually store these images and become excellent students when they come to the university. As it is now, we have to start from scratch, from zero, when they come to the university, and then we very rapidly increase the learning curve, but it would be perfect if they had some visual understanding of chemistry when they start. So with that, I would like to end this and thank you very much for your attention. And also I would like to thank all my colleagues. In particular, I would like to thank Christian Tornø, Claus Bock, who was my mentor, and Michael Bolz, who invited me to University of Copenhagen and opened up all the opportunities there. I'd also like to thank the Danish National Research Foundation and Carlsberg Laboratory and University of Copenhagen for the support over the years. And not the least, I want to thank all my colleagues. All of these people have had something to do with the development of a click chemistry in our laboratories. And my family, Phaedra Marie Saint-Hilaire, Gianni Oliver Saint-Hilaire Meldal, Anna Davolio Meldal, and Sandra Davolio. Thank you for your attention.
Thank you for that fantastic, animated and engaging talk. Thank you so much. Okay, cool. So it's time for our third laureate, Barry Sharpless. Barry Sharpless was born in 1941 in Philadelphia, Pennsylvania, USA. He was awarded a PhD from Stanford University, California in 1968, and is now active as professor at Scripps Research, La Jolla, California, USA. So please join me in welcoming Dr. Sharpless up on stage.
Introduction and ladies and gentlemen and friends and former students and colleagues out there too. It's a unique feeling. It's awe-inspiring to have two lectures that I, of course, understood every word of, but also that I couldn't have given myself because they are so beautifully organized. And That's my failing in life. I can't plan things. So some things need to be fixed up in the final written version of this. But I have some observations to share. What I really like to share is how the three of us were wisely chosen by this committee because we have the same spirit that we've evolved from our experiences over, well, in my case, a longer lifetime, but it's a profound thing to realize what you can do if you don't try to just do any old way of making molecules. And that's the main message I can give you. So I don't know, is this a forward and reverse thing too? Or okay, so yeah, this is the launching of click chemistry, and it started before there was any really motivation for knowing that it would work, but I did know that some reactions were better than others. And anybody who's been a chemist for years, like Morten and I have, know that you don't really have much faith in a reaction that's never given you more than 10-20% yield. Why would you consider using it in a plan? So even if you're planning your synthesis very carefully, there's only about a handful of reactions that good chemists would know, okay, I can go to the bank with this one, I think. Here's the way it started in, let's see, how do I push the laser pointer? Oh, it's right there. Okay. So this is where click chemistry started, in this old building which had pigeons in it and stuff, and we cleared it out and built some labs in there with Alfred Bader's funding. Alfred and Isabel Bader, well, Alfred, he was coming around to Dartmouth College even, and to Stanford with his patched elbow suit and his handing out coffee cups. And our generation knows who Alfred Bader is. Younger people, like I'm sure Carolyn never ran into him in that role, but he was the man who said, "What can I do for you?" Now, here was a man making chemicals for sale, and how many would go around door to door like that? But that's the way he was. And he started his lab in a garage in, I forget which city in the Midwest. And he was an important man who decided, and Victor Sneakus at Queen's College in Canada, he's indebted to Alfred too, because he endowed a big chair for Victor, who was another wonderful chemist who I've admired for years. And Hartmut Kolb is here in the audience, and Hartmut was the first click chemist because he came back from Ciba-Geigy when it merged. Had been a postdoc in the lab. He came back from Ciba-Geigy when it merged with Novartis, with whatever the other guy was, Sando, and became Novartis. And when something like that happens, the quality's going down. And Hartmut got out of there wisely, and he started organizing a chemical group, and he made hundreds of blocks, building blocks for chemists to have versatility in discovery chemistry. They thought these blocks were actually supposed to be small drugs or something in Japan. It just was ahead of its time. It didn't have any precedent for working. And so people didn't know what we were trying to sell. And somebody bought the company to make it into a drug supply line for their own new startup pharma company. So Hartmut left again. And okay, now this is the man who made it possible for click chemistry. It may not be known to many people, but I couldn't get funded very well because I don't write grants very well. And I could never get funded for doing click chemistry. They hated it at the NIH and the study sections. They wanted total synthesis or complex very complex hammer and tong ways of making big molecules in those days. So Richard rescued me, saw my potential, and he funded click chemistry. 90% of all the funding for click chemistry came from Richard's rich friends, and that he could constantly get to support his institute. My wife, on our 5th anniversary, well, she's the genius in the family. I won a Nobel Prize, but everybody knows she's the genius, and she names things like click chemistry and Sleeping Beauty, but her click chemistry is always stuck, and it stuck in the craw of a lot of people for years because it wasn't popular. It just didn't appeal to people. It seemed Mickey Mouse, and anyway, I can't say that I would be here today, or I would have been here the last time either, if it weren't for Jan. And I don't know where anybody's sitting. I get nervous, so I'll just say, oh, good, over there. Okay. Yeah, she keeps me connected to the real world. And besides also having really incredibly, she's a writer, and she's trained English major writer. Now, is this? I guess I don't know how to change this. Let's see. How do you make this? I can't get the normal thing to. What is it? Maybe I should start it again. Just go back and try to do it without the comments. Okay, then I go. Let's just go this way and see. No, it's going to go that. Okay. There. Oh, wait a minute. What happened? So I was born to be a process chemist. I was a fisherman from Norwegian, Swedish fishing family. My grandparents, and I worked on fishing boats until I was ready to go to college, and actually even while that time, before I went to Stanford for graduate school. And so I like catching things and interesting things, and also quantity was important when you were fishing for commercial reasons. So a process chemist, I'm not interested in little tiny scale reactions. I mean, I can do them and understand the meaning of them for analysis, but I want to make a handful of something, and so that I can do something, whatever I want with it, and just put it on the shelf as a reagent. So that's what a process chemist does. And so simplicity, my lifelong mission was to make things faster. I'm very impatient in the lab, and my son Will, he's here, who worked in the lab a year and a half, he's like me. We make things quickly, and they crash out, and we go on. And so the asymmetric oxidation got me interested in well, this is not quite written the way I would. The main thing I learned came from George Hammond, a nonlinear thinker who was a professor and came back to work at Exxon for years. It's not making compounds that we're interested in. There's no sense to making compounds. There's too many of them. But it's the making of properties. You got to find properties. That's the magic of chemistry. We have properties all around us we haven't made yet, things that are easy to make. And that's what click chemistry has helped. Yeah. Now, this is why I'm disorganized probably, but the periodic table was very helpful to me. I dove into it as a kid in high school or even before in grammar school, and I read all about every element and its properties and does it react with water, or I just needed to know about everybody. And of course, a lot of these things you can't find anything out about. You go and look for things here you won't find. Well, cerium, these are okay. These are important today, and countries fighting over them. But the transition metals really set us off during my lifetime. But, you know, I'm not so enamored of them right now. And copper, of course, is in Never Never Land here between the main group and copper is really, really special. It's very slippery. You can't stop it. It's everywhere in our body at attomolar concentration. And so there's plenty of copper if we can learn to house it better to do the chemistry that Carolyn and Morten would like to do even when the heart is beating. Elements in the table that I particularly have a relationship with is selenium. That came for an interesting reason. MIT, I thought I needed to write an NIH grant because I didn't think NIH grant would support chemistry. That's how naive I was. So I went out to Wisconsin for 2 months, learned from the experts on selenium biochemistry, came back, wrote an NIH grant, and then proceeded to discover that selenium was fascinating, simple chemistry, making olefins. So in 2 years of doing that on the side, it got me tenure at MIT. Okay. So Derek Barton was a friend and advisor to me. I liked his way of looking for new reactivity. And I was actually told by him I couldn't stop doing asymmetric chemistry if I wanted to win a Nobel Prize, which I thought was, that was Derek's point of view. And I was looking to get out of it because I can't stay on something very long. But went on for a while. Hartmouth came and we really did a job on the asymmetric epoxidation, dihydroxylation. That was probably the best reaction I've ever seen for catalytic oxidation. I put this in for those who, I'll leave it in the written version, and it just enumerates the events. Epoxidation was fine, and it sidetracked me with chirality. But chirality wasn't what I was interested in. I was interested in making efficient bonds. And I found out more and more the only bonds that matter are the bonds between things that are separate, like people. The first they get in the same room, maybe they hold hands, that's like Velcro. But then eventually, if you're wearing the right bracelets, there's a click, and then you're together forever. But when you put two things together, molecules, modules, that's where the properties come from. That's what nature teaches us. But we don't have the right to build things the way nature does, top down. She's making big molecules whose sole reverential involvement is to control themselves and the other guys to make the right connections. And so, no, but we can start at the bottom. That's our privilege. We start at the bottom with a few things. Maybe we need a few thousand because we don't have all this massive territory we can cover, but we can come up from the bottom and make function. And that function can be affordable for hopefully much more people than biotechnology could possibly imagine being. Anyway, I made some enemies in this world. Enemies came heavily from saying the grail of carbon-carbon bond formation was perhaps a false grail, and I would defend that to this day, but let's not get involved in that. That probably set back click chemistry's acceptance for years. And MG came, he was a great student during the asymmetric epoxidation and was the man who did the mechanism at MIT. But he was in Virginia, and then Richard Lerner loved MG when he was out on sabbatical, and he hired him as a professor at Scripps. And then MG and I got together, and we had, let's see now, like, I'm just going to jump to the end whenever I get in trouble, so don't worry. Overnight, so Richard, he just Richard loved this project too. It was the idea of, look, why don't we let the enzyme take 50 pieces on each side and do the cycloaddition, the azide Carolyn's thing, where you get this very weak, Weiskin, our hero, all of us, because of his great, physical organic chemists are much more important than people realize, and we didn't do them much good in the last 50 years, so I suggest we worry about them again in the future to help us. But so the enzyme can hold this thing together, and this cycloaddition could increase its likelihood. But it seems very pathetic because the concentrations we had of the pieces, and you'll see them in the next slide, this is what MG and I decided to do. Oh, that's our first article on the click. That's the manifesto that was written for click chemistry when we didn't even have a perfect reaction. So we were kind of making it up as we went along here, what might be. But we didn't have those reactions at that time. And where's my, no. Okay, this inspired MG and I, and Reza Ghadiri, a beloved colleague at MIT, bought a copy of Kevin Kelly's book, Out of Control. If you haven't read Kevin Kelly's book, you've got to read it. Even though it's written in 96 or 94, it's still relevant today to what life is, that a lot of us don't think of about life. And life is out of control. Of course, naturally, if it were in control, who'd be controlling it? And it would fail if it had some human controlling it. So Kevin Kelly says, nothing is more addicting than God games. He's got a chapter on God games. And don't get me wrong, these God games are not irreverent. Sim Earth, Sim City, whatever. But your first rule about God games that you learn is to be a God, you have to relinquish control. You set down your system and you watch it. Well, I mean, those games sound really hard to imagine succeeding. And so we were out walking on the beach at Scripps a few days later after reading the copies of the book Reza gave us. And we both said, well, what did you make of this book? What did you like? And we both loved the God game part because we realized there was a question where you could really see a problem. Because everybody takes x-rays of big molecules and they're sitting still, frozen to death with their mates at minus hundreds Kelvin. And then we make predictions about how we're going to stick something in this hole and other things we take. But it's like the ugly stepsisters trying to get into the Cinderella slipper. You know, they're breaking everything. And so humans are trying to design the finished product. Oh, you're stable in water, you could be a drug. That's really arrogant when you think about it. So what about if you let the molecule take a couple parts and somehow it can make a connection that's permanent? So the molecule can act as a reaction vessel, and that's well, chemical space is big. This is something that everybody knows about today. I give Derek some credit here. I should mention Regina Bohacek, Colin McMartin, and Wayne Guida, who also had an elegant story on this, how the mass of the universe that we can see, the visible universe, could be occupied by one molecule of every possible drug that was less than 500 molecular weight. And it wouldn't be any matter left over to work with. So it's absurd how much stuff there could be. So my guess is about 99.99% of space close to us, 2 or 3 steps away, 4 steps away from things we could execute to have good, perfect reactions that could make a useful molecule strike into a real functional molecule. But that the screen is the problem. This is where I would say we should, that's my insight from the last few days. I thought, you know, the biggest problem we have is screens. I mean, how are you going to discover, I have to stop, right? 10 minutes, okay. Well, we got some extra time from the failure, okay, thank you. So how are we going to screen for something? I don't know what you want out of a chemical. Everybody might have their own magic chemical interest for function from a human being or an animal. But it's how do you screen for that? You're not allowed to screen directly in humans legally, so that's a problem. But there are other things too, like how to make a building just overall have something that makes it last 100 years longer than wood for falling down. Corrosion in concrete, or you'll see what happened to me. Most of the chemists, we think about life, especially since Berzelius put us into thinking about life as organic chemistry, which was a very misleading thing and absolutely absurd, of course. But that's okay. He did some great things for chemistry, but his own student Wohler proved him wrong, of course. From Germany, a postdoc. So here's the in situ with fancy Mike Peake at Scripps could make anything look great. Richard Lerner loved him. And this is the enzyme acetylcholinesterase. And here it is making in its guts this thing that MG and I worked on together, making the triazole, clicking it together in its heart. And the inhibitor that came out was I'm going to have to jump over this one because what was the inhibitor? Why? Okay, maybe that'll come later. But the inhibitor was femtomolar, and it went down as low as 2 femtomolar. I mean, and this was the most potent inhibitor ever made from almost any enzyme, but let alone for acetylcholinesterase. So that was the first inhibitor we ever made in my lab, and it was made by MG and Warren Lewis and Luke Green. That was femtomolar. And that's pretty remarkable, from 50 pieces on each side. So there were a lot of combinations. The certainty of molecular chance. Well, when we started doing this assembly of modules, we found out we ran head-on into a problem. The best reactions that were out there were far from good enough. And here, I'll just show this slowly, quickly. Here's the quickest way we found, MG and Hartmouth and I working, and Jan in the last few days to figure out, because we had other ways of explaining this. But here, if I have a perfect reaction, what does it look like? Well, it actually looks like this. After 1,000, you can do this in the computer. You find this thing, you put it in, and it takes it to the power from 1 up to 1,000 or higher, whatever you want to do. So here it is. If you do 1,000 steps in a row with a 99% yield, and most organic chemists are going to go to the bank with this kind of a reaction. And in solid phase synthesis, Morten could tell me they don't need that even, they just use excess reagent, right? And so 99% yield, this is what it buys you after 1,000 serial steps. You're down to 0.004. And then I come to 99.9, I get up to here. 99.99, I'm here. But to get to 99% after 1,000 steps, this is polymer chemistry, right? And here you have 99.99, usually helps to have another 9. And the thing almost never fades. And that's what this new reaction is like. And it's a polymer reaction. So, okay, let me just get through to that. This is the reaction that's magic and came into our lab with Jia Dong. It's a suffix reaction. What happens is silicon, phosphorus, and sulfur love to change ligands with a suffix catalyst, and they do it as fast as the QAC reaction, if you can believe it. Room temperature, 130 degrees, it doesn't matter. The reaction doesn't even explode when it's done neat. It's a polymer reaction. And here is the early team that published that. And Ponmi's been working for a long time with me now at Scripps. He came back he was a student who came back as a professor at Scripps, and we work on the polymer together. Here's MG, who's been involved in suffix chemistry and click chemistry from the beginning, almost. Hartmouth, of course, was really at the very beginning. Here's Jia Dong and Larissa Krasnova on this suffix chemistry. Now, what I want to show you is just how the polymer, you melt the polymer together, the two monomers crystalline, melt them together, and then you put the catalyst in, it makes the polymer, you put some more DMF in, you put DMF in, and then you pump it into a stirred solution. Jia here is showing, you get it crashes out in methanol, you wind it up, he called it bird net soup, bird net polymer. It's a beautiful polymer, very, very good fidelity. Okay, I know I'm running out of time here, but here you can see, no, I have no time, right? Oh, okay. So 500, here's another type we make from another gas from sulfur, the tetrafluoride. And this polymer is very, very easy to make. And it forms perfectly, and it has this extra spot where we put AZT out there with a connection through a QAC. And so we had this huge molecule with 500 thymidines on it. Every spot had a thymidine sticking out, and it was a helical structure. And if we didn't have this yield up here at 99.998, we wouldn't have gotten this yield. I'm sure this yield's too high, but even if it was 98, you can see the massive amount of yield you get out of this reaction. And here's the NMRs. The NMRs are ridiculous. This is a molecule with 500 units connecting, and this is the proton NMR, and this is the reference for fluorine. That's the fluorine at 500 megahertz or whatever the resonance frequency is, but it's just like one fluorine. And these got chiral centers in them, and they're helices. Here's what the thing looks like. Han Zulaf is our collaborator in the Netherlands, and his students got these pictures of these helices. The pitch of some of they know the pitch of them. Each of these will have like a nucleotide sticking out every turn. Its pitch is about 12 degrees. But the main point is that this is DNA, of course, and that's another nice polymer with sulfate, phosphates, affinities. But when you look at this from outer space and you come in and you're smart, you're looking at somebody shows you that, you'd say, well, those are phosphates or sulfates. You can't tell by looking at them because they're either neutral or they have a -1 charge. Just to remind you that this chemistry that's helical is also related to life's chemistry. And finally, this is what I wanted to say. We ran head on into an amazing thing. We've been trying to do biochemistry, but I never did polymer chemistry before, and I found it very hard to do. I didn't understand the arguments, and we got tremendous help from Molecular Foundry at Berkeley. And Carolyn used to run that Molecular Foundry. Can you imagine? She does she's a jack of all trades, I guess. But Yi Lu is a former student with MG and I. And Yi has been there for 10 years or so. And Pong knew him, and we got going. They would measure our polymers, our polysulfate polymers. And the main thing about these polysulfates is they are immortal. As much as you might say a sulfate looks like a weak wimp link, this thing is not coming apart in vivo. It's just stable. So it's just a connector. And here's what it does. If you make this polymer and then you use it to make these films, you just dissolve it in a solvent and make films, and you put those films on as capacitors. This was Yi and his student Henry's idea because they had experience with capacitors under duress, under high heat and high temperature, and we don't have good ones right now. So what you make is sort of wrapped up coils of these films, and you wrap them around the zone where the horrible things are happening in a Tesla or a power station. If you have a capacitor, it has to load its charge and then wham, drop. And if it leaks, and you get heat, and you get so much heat that you can't function anymore, and the high field doesn't help either. They're both against you. So what we found was something amazing, I thought. This is one of the polymers we made, and we made it to have a high TG so that we could have a high temperature. And these are the gold electrodes, and the film that's laid down on those is our polysulfate film. It's clear, and then you wrap it around in so a way that you can run the field through it, and at 150 degrees, 750 volts, it gives the polysulfate is way up there ahead of all the ones used in electric cars now, or for any purpose where you need survival of this electric sort of, it's an insulator, but it holds the charge and then it drops it. Loads drops. So we ran it 50,000 times over 3 hours, and it just runs straight. There's no decay, and if we want, we can show that. They showed that if you wound it, then it makes a short, and then you do a few square wave cycles, and it comes back on a reproducible cycle, and it loses maybe 1 or 2% because it healed itself. It just burned up the part that wasn't any good, and now it's back to being a healthy capacitor material. So this was a complete intrusion into my life of physics, and I thought that was really a cool thing, because how physics, especially physics with fields, right? Electromagnetic fields, this is magic. It's action at a distance. How the hell is this power? Well, we know, we think we know, and how it's working. And we're after that now. And a man in Germany, Dietmar Stahlke, who's one of the few people I think understands main group chemistry, bonding, aluminum, silicon, sulfur, and phosphorus and sulfur. That's really messed up, that bonding, as it stands now in our life as chemists. So his way of looking at it, he can explain why this thing is not conducting. This phosphorus-sulfur linkages will resist conduction. There are no pi bonds. Most people, we all draw them as pi bonds. Oh, pi bonds. There are no pi bonds there. So that's the end, but I just had to get that last message out, that the certainty of chance, this is the certainty of chance that Jan wanted me to introduce, because I just, I can't get over how much chance, when you can make things quickly and you can test them against something reasonable, the certainty of chance is huge, right? I mean, do you intuit that? That's why I think it's important, that certainty of chance. And that's from a guy who is a jazz musician, and the first person to say that he believed in the certainty of chance. He was I can't remember the Dadaist type movement, and the fact that life is one of the chances that we see here in front of us, is one of the chances this universe offered, and it's occupied. Sorry for all the lack of order, but I just thought, humble thanks to all my wonderful students and postdocs and visiting professors and collaborators, and I'm going to get the names out there, of course, in a long list in the paper, but I couldn't get them together for today, so I apologize. But the ending was the main part of the story. It's certainty of chance again. Thank you.
Stay. Stay on stage. Okay. So we should stay up here. Okay. Yeah. Thank you so much, Barry, for that fantastic, awe-inspiring talk. Right? And may I now call back Dr. Bertozzi and Dr. Meldal to come back on stage so we can all thank all three laureates for their fantastic lectures and accomplishments. You should come up. It's